Okay, now we're going to cover sections 3 and 4 in chapter 9. And here we're going to talk about cancer, which is as is shown here in its earliest stages, is essentially an abnormal growth of cells or neoplasm that begins to form a tumor. And in some cases they can spread and basically cause all sorts of problems. So normally cells are controlled in when they divide, but cells that have mutated and become cancerous just grow and divide uncontrollably. And in particular, we're referring here to the malignant kinds. You can get benign tumors or benign growth, and those are ones that tend to grow very slowly and not spread. But malignant ones are ones that have the ability to grow much more quickly and the potential to spread around the body. So let's make a comparison here. So normal cells become differentiated. That is, they have a particular job they do. They might be muscle cells, nerve cells, etc. But cancerous cells remain in this undifferentiated state. They do not become a muscle cell or nerve cell. They're just a cancerous cell, and they don't do what those other cells do. They just simply divide and take up resources. They have mutations, that is, their nuclei are not normal, and those mutations are what are causing them to divide uncontrollably. Normal cells, when they are damaged, will often go through apoptosis, or if they've just reached sort of the end of their lifespan, they will go through apoptosis, but this doesn't happen with cancerous cells. Normal cells have what we call contact inhibition. That is, they grow to a certain extent and are in proximity to other cells, and that proximity then inhibits them from growing anymore or dividing again. But cancerous cells lose this contact inhibition. That is, again, those cells, that clump of cells will just keep growing, keep dividing, regardless of how crowded they get or how many are there, and they just pile up and form these disorganized, multi-layered tumors, whereas normal cells have an orderly pattern and they sort of stick within that pattern. Cancerous tumors will spread or metastasize, and they stimulate the growth of blood vessels into the tumor to supply food, oxygen to that tumor. Angiogenesis. So indeed, metastasis is when cancers spread throughout the body. Cells will dislodge from a tumor and they can spread through the circulatory system, or this other thing called the lymphatic system. And you may have heard of lymph nodes. You have them throughout your body. It's not uncommon for a metastasizing cancer, those cells to spread to lymph nodes and get deposited there and begin forming new tumors. Again, angiogenesis is when the tumor stimulates the growth of blood vessels into the tumor. Well, so cancers get started when you have a mutation or a change in the DNA of a particular cell, such that the DNA is no longer giving the proper instructions to that cell, you might say. And this is a particular problem when you get these changes in what are called proto-oncogenes. Proto-oncogenes are ones that control the cell cycle or cell division. And there are also genes that are responsible for controlling the cell cycle, but in particular they slow down or inhibit cell division. And so what can happen with these mutations is when, for example, a proto-oncogene mutates, it can become an oncogene, and so that gene now basically causes those cells to become cancerous and form tumors. And when tumor suppressor genes are mutated, there's no particular new name for them, but when they mutate as well, they can also lead to the same outcome. So essentially, again, these proto-oncogenes, they're normal genes. They control the cell cycle, but when they mutate, they can become oncogenes, and they basically overstimulate cell growth and division. Don't worry about this image too much. It's out of the AP book. Just focus on this part, really. Things that can cause these mutations: radiation, certain chemicals, viruses can spread. When they infect a host cell, they basically inject their DNA, and certain viruses are known to, when they inject their DNA, to cause problems and promote cancers. You can also inherit genes that have mutations on them, and so certain types of cancers are known to be hereditary. So in normal functioning cells, you have certain compounds, these growth factors, that stimulate the cell to proceed with the cell cycle and divide. Okay, when this growth factor is not there, it won't do that. Well, sometimes what can happen is, so in the cell, there's a series of reactions that occur, a series of molecules involved with getting that external signal basically into the nucleus, and making use of the instructions in the DNA to go through cell division. Well, what can happen is, if you get mutations in some of these compounds are going to typically be some kind of protein, and proteins are basically made using the DNA, and so that mutation in the DNA leads to a defective protein, and if it happens to be one of these proteins involved in controlling the cell cycle, like these tumor suppressor genes that make these proteins that control the cell cycle, then you can get uncontrolled cell division and cancer. So again, proto-oncogenes, tumor suppressor genes, when that DNA is defective, it can lead to defective proteins, and the cell cycle is no longer properly controlled. Here's a well-known example of some of these, an example of a gene that in a certain form can promote cancer. And I have a picture of Angelina Jolie here because she made news recently because she got tested and knows she has a form of, I'm not sure if it's one or both, but basically a form of these BRCA genes, as they're caused, that gives her a very, like an 80% chance of having breast cancer. And she, of course, inherited this from her parents, and I believe her mother had breast cancer. And so she made the decision to have, I believe, a double mastectomy, which essentially removed the breast tissue and reduced her chances of getting breast cancer significantly. You can see this BRCA1 gene is found on a particular part of chromosome 17, and again, it's a gene that helps control the cell cycle, and certain versions, mutated versions of this gene, give you a greater chance of having cancer. Okay, so now the fourth section covers cell division in prokaryotes, fairly short section, and so bacteria, prokaryotes, have a simplified form of cell division compared to eukaryotes. They do what's known as binary fission. You'll recall that prokaryotes have relatively simple cells. They don't have a nucleus. They only have a single chromosome. They don't come in pairs. They just have one. And it happens to be circular and not linear. And so the way they do cell division is, first they make a copy of that chromosome. The chromosomes, the two individual copies kind of attach to ends of the cell, and the cell sort of elongates, which essentially moves the chromosome to the two copies to opposite ends. The cell splits in two, and so you end up with two bacterial cells that are clones of each other. Prokaryotes do not do mitosis. Mitosis only happens in organisms, in eukaryotes essentially, that have a nucleus and multiple chromosomes, and so it's not needed in prokaryotes. They simply make a copy of the chromosome, kind of elongate, split in two, and there you go. Whereas cell division in, well, here, I think I got a table down here. Yes, no mitosis. So let's look at the differences here. So prokaryotes, again, they do binary fission, and they're just doing this to reproduce, a form of asexual reproduction in which they're basically producing clones. Now, cell division in eukaryotes, in some organisms, it's done for the reasons of reproducing, particularly certain types of single-celled organisms, protists and yeast, for example. But in more complex multicellular organisms, it's mostly done for growth and repair. So those are some differences in the functions of cell division in prokaryotes, eukaryotes. Again, cell division in prokaryotes known as binary fission. In eukaryotes, cell division takes advantage of mitosis, the division of the nucleus, which again does not happen in prokaryotes. Okay, there we go.